Hello students. Today we start the somatic embryogenesis. So the somatic embryogenesis is nothing but the embryo production. So in flowering plant, this is the one of the characteristic features of the flowering plant, that is embryo production. So somatic embryogenesis is nothing but the formation of embryo-like structure on the cultural callus tissue is called the somatic embryogenesis. That means from the callus there is the formation of embryo, and this process is called as the somatic embryogenesis. So embryoid, that means the embryo-like structure on the cultured callus tissue is called as embryoid. So from the callus, after the reculturing of the callus, there is the formation of root and shoots. But the callus will develop the embryo-like structure that is known as the embryoid, and this process is known as the embryogenesis or somatic embryogenesis. So there are two types of somatic embryogenesis. First one is the direct somatogenesis, and second type is the indirect somatogenesis or somatoembryogenesis. So let's discuss one by one. So first one is the direct somatic embryogenesis. So in this process or in this method, the somatic embryo are raised directly from the explant tissue without any callus phase procedure. So for example, the citrus plants, you have to see in this flow diagram. So if we select the immature green citrus fruit, after the surface sterilization, the surface sterilized with the mercury chloride, then remove of the ovules of these fruits. Then ovules will be separated from the nucellus by cutting the ovules. Then nucellus is and then again transferred on the MS medium. Then from there the somatic embryo will be formed. Then this somatic embryo is again transferred on the medium or transfer on the agar medium, so that we can got the plantlets. These plantlets are transferred after the sufficient development of root and shoot. These plantlets will be transferred from water plants. That procedure is known as the acclimatization, and then these potent plants will be slowly hardening, and then the fruits are harvested from these plantlets. So this is the entire process of the direct somatic embryogenesis. In that, shortly you have to remember that in immature fruits, the ovules will be separated from the nucellus. Then this transferred of the MS medium, and from this nucellus on the MS medium, the somatic embryoids are formed, and this embryoid again transferred on the MS medium, so the plantlets will be formed, and these plantlets will be slowly acclimatizes or hardening, and then the whole plantlets will be obtained, and on these plantlets fruits will be developed. So the immature green fruits of the citrus are selected for the somatic embryogenesis. Then the fruits are properly washed with soap and clean distilled water to remove the contaminants from the surface of the fruits. Then ovaries are separated from the fruits with a sterile knife. The ovaries are surface sterilized with 2.4% sodium chloride for 15 to 30 minutes. The ovules are removed with a sterile knife. The nucellus tissues are removed from the ovules by sterile scalpel. The entire nucellus tissues are inoculated in a flask containing MS medium or and white vitamins. Then the culture flask is incubated at 25 degrees centigrade to 29 degrees centigrade with 1000 lux light for 2 to 6 weeks. During incubation, the nucellus tissues undergo differentiations and develop into the embryo. The somatic embryos thus produced and transferred to agar medium to grow into the plantlets. Then these plantlets are hardened, acclimatization, and then potted. So this is the process of direct somatoembryogenesis. In that, first of all, you have to select the immature green citrus fruits. Then these fruits will be sterilized by the or they will be washed with distilled water or soaps. Then with the help of knife you can separate the ovaries from these fruits. Then this ovary will be again washed, sterilized with 2.4% NaCl for 15 to 30 minutes. Then these ovules with the help of knife, the sterile knife, we can separate the ovules from the nucellus. Then this nucellus tissue will be again sterilized and it will be transferred or inoculated on the nutrient medium flask or with the help of some white vitamins. Then these inoculated flask will be transferred on the incubation chamber. Incubation chamber, that means the room where light and temperature will be controlled in sterile condition. Then these nucellus develop into the somatic embryos. Then the somatic embryos are again recultured on the agar medium. So from these somatic embryos we can got the plantlets, or they will be developed into shoots and roots. After the sufficient development of shoot and root, they again retransferred into the pods. Then they will be slowly hardened and acclimatization, so they can potted into the, or they can transferred into the pot. So this is the direct somatoembryogenesis. Then second one is the indirect somatoembryogenesis. Indirect, there is the production of indirect somatic embryo. Example is the carrot. So in carrot seeds, we can take the carrot seeds. These carrot seeds are surface sterilized. Then they will be transferred on the MS medium. Then we can obtain the seedlings of these carrot plants. These cutting the petioles of the seedlings, then again it retransferred on the MS medium with supplements of the 2,4-D. Then from that it will be obtained the callus. This callus is again transferred on the MS medium with supply of the 2,4-D. So we can got the somatic embryo. Then this somatic embryo is developed into the plantlets. These plantlets again potted plants, and we can got the entire carrot plants. So, in indirect embryogenesis, the carrot seeds are sterilized by the sodium hypochlorite for the 50 minutes. Then these seeds washed with the sterile distilled water to remove the sodium hypochlorite. The seeds are inoculated on solid MS medium and incubated at 25 to 29 degrees centigrade, then 1000 lux light, about 16 to 24 hours. The seeds start to germinate within 2 to 6 weeks. The young and small petioles of germinated seedlings are removed by the sterile knife. It is transferred to MS medium containing the 0.45 mm to 4D. Then the culture is kept on incubator at 20 to 25 degrees centigrade for 4 to 8 weeks. During the time of incubation, the petiole of explant produce callus tissues in the nutrient medium. So this callus can be subcultured in fresh nutrient medium containing the 0.45 mm to 4D and incubate at again it incubate at 20 to 25 degrees centigrade. The callus tissue thus produces shows embryos and embryonic initials which develops into plantlets in course of time. The plantlets are transferred to greenhouse for proper growth point. After proper growth of plantlets are transferred to the main field of planting. So this is indirect somatic embryogenesis. In that we can use the carrot seeds. These carrot seeds will be sterilized with the sodium hypochlorite. That is transferred on the MS medium. From these seeds we can got the seedlings. Then cutting the petioles of these seedlings, and again they will be recultured on the MS medium with supplements up to 40. So we can got the after incubation period we can got the callus. So these callus are again recultured on the MS medium with the supplements of 0.4 mm to 4D, so that from this callus we can there is the development of somatic embryo. So this somatic embryo, after the incubation period, they will be developed shoots and roots, and we can got plantlets. So these plantlets will be transferred on the pot, and they will be slowly hardening with the help of greenhouse. So this is the indirect somatoembryogenesis. So today's lecture I have discussed about the two types of somatic embryogenesis. First of all, this somatic embryogenesis is nothing but the formation of embryo-like structure uh, on the culture of the cultural callus, that is somatic embryogenesis, and the embryo-like structure that is known as the embryo. So these somatic embryogenesis are two types. First one is the direct somatic embryogenesis, and second type is the indirect somatoembryogenesis. Direct somatoembryogenesis example is the citrus. In that from immature fruits we can remove the ovules. These ovules again separated out the nucellus, and these nucellus are transferred or inoculated on the nutrient medium So we can got the embryoid, and these embryoids will be develops into the plantlets. And in the indirect embryogenesis, we can use the carrot seeds. These carrot seeds will be sterilized and grow on the nutrient medium. So on this nutrient medium, seedlings will be developed. So with the help of knives, we can cutting the petioles of the seedlings, and they will be recultured on the culture medium. Then callus will be developed, and from this callus again transferred on the nutrient medium with the help of supplements of the 2,4-D. So can got the somatic embryoids, and again these somatic embryoids will be recultured or inoculated. So can got the somatic embryoids. The somatic embryoids are developed into the plantlets. So this is the two types of the somatic embryogenesis. So next we have talked about the commercial aspects of somatic embryogenesis. So from small tissue many plants are produced. The plants are showing all superior characters as that of parent plants, that is transferred to the parents. All plants raised by somatic embryogenesis are identical, not showing the variations. Then any somatic cells develops embryoids and embryo every time, which is rare as compared to the somatic cells. So the plants raised by somatic embryogenesis are disease resistance and embryonic viability will be increases. So this is also the important applications of the plant tissue culture in the somatic as a somatic embryogenesis. So we can got the number of plantlets from these techniques. These plantlets will be superior, disease free, they not showing any variations, and also they will be uh, improve the embryonic, or they will be increases the embryonic viability of the plantlets or the plants. So this is all regarding the somatic embryogenesis. So now I stop here.